Thank you, Lionel, for inviting me to present at the 4th annual workshop. It's hard to believe that this is already the 4th workshop. It feels like you just started this. And today I'm going to talk about autofluorescence lifetime imaging that we did in our lab to study cell division. And so cell division is a series of events that includes DNA replication in the S phase. Let me bring my laser pointer here. And then the newly replicated genome will then divide into 2 daughter cells in the M phase. And then these 2 phases are separated by the gap phases. So G1 is the gap phase between M and S, where the cell prepares for DNA synthesis. And then G2 is the gap phase between S and M, where the cell prepares for mitosis. And these phases, cell cycle phases, are exceptionally well preserved in the eukaryotic cell cycle. Now, these are all complex processes with high energy demands, and it is not completely well understood how the cell would regulate the generation and consumption of energy during all these processes. There are several research that has correlated metabolic pathways like glycolysis, oxidative phosphorylation with the cell cycle machinery. And it is this connection between metabolism and the cell cycle that if it's altered, then it will cause different pathological conditions, for example, cancer. And then manipulation of these connections will help us come up with better designs for therapeutic strategies for all these diseases. There have been many previous researches in trying to understand metabolism and cell cycle. This is an example where the researchers looked at NAD plus NADH ratio in HeLa cells. And they found that it fluctuates at different cell cycle phases. And particularly, they found that this ratio decreases during the gap phases and then increases in the S phase. But they did not have any, they could not measure this during the M phase. So this is because the temporal resolution of the methods they use were not good enough to actually capture M phase, which has the shortest duration among all the phases. And moreover, all these techniques, they look at bulk measurements, so they cannot pick out single cells and look at metabolism of that cell during its cell cycle phase. So this led us to the aim of this project, where we want to develop a non-destructive tool for label-free imaging of cell division. And more importantly, we want to do that at single cell level. So as Lionel mentioned, we exploit 2 fluorescent molecules. These are endogenous molecules which are already present in the body, NADH and FAD. And these are also important coenzymes that are involved in oxidation reduction reactions in key metabolic pathways. So for metabolic imaging, we look at 2 parameters. One is the optical redox ratio. And we define the optical redox ratio as the intensity of NADH divided by the intensity of FAD. And this ratio provides a quantitative measurement of relative oxidation reduction states in the cells. So an increase in this ratio is indicative of a reduced redox state, while an oxidized redox state will cause decrease in this ratio. And then the other parameter that we look at is the NADPH lifetime, which you have already heard about. So the free NADH has a shorter lifetime because of its folded conformation, and then bound NADH has a longer lifetime because of its open conformation. And the lifetime can be estimated by fitting it to a double exponential decay. And then we can calculate the average lifetime, which is a weighted average of tau1 and tau2, which is indicative of free and bound lifetime. So basically, lifetime tells us about the enzyme binding status of NADH. And then to obtain all these values at single cell level, we do single cell analysis, single cell segmentation. So for that, we first take the NADPH intensity image, and then we use CellPose to do automated segmentation. So CellPose is a deep learning-based segmentation method, and we can either use models provided by CellPose like the Cyto2, or we have also retrained their models with our data. So once we have the cells segmented, we take the output of CellPose and we do a manual check. So we can check whether the cell boundaries have been correctly identified. And we use Napari to do that. So we can basically load the NADH NADH intensity image and the mask from CellPose. And then we can manually check. And then also we can go ahead and edit if we are not happy with the masks. And then the output from Napari is the single cell mask, which we can then apply on our autofluorescence data to obtain single cell redox or single cell lifetime. So for this project, I have also acquired time course data. And for those data sets, single cell lineages were tracked on TrackMate. Then the time course tracks could be aligned. So we aligned it based off of the time frame when the first daughter cell appears after cell division. Okay, so I have divided my talk into two parts. First, I will show some of the results of autofluorescence imaging to study cell metabolism during somatic cell division. And secondly, I will talk about our study on early embryonic cell division. So for somatic cell division, we looked at two commonly used cell lines, the HeLa cells, which are cervical cancer cell line, and then the MCF10As, which are normal epithelial cell line. They also have different doubling times. So these are some of the imaging parameters. Most importantly, we made sure that while we are collecting the data, the cells are at 37 degrees and 5% CO2 so that they're alive during the whole imaging process. And then we did sequential imaging of NADH and FAD, and we acquired data every 20 minutes. So then to calculate the optical redox ratio at single cell level, we segmented the cells, and then we followed each cell per time point and calculated the corresponding redox ratio for that cell for each time point. So here I have represented that as a heat map. So each row here is a single cell, and then each column here is a time point. And then I divided the cells into two groups, so dividing or non-dividing. So basically non-dividing is the cells which did not divide during the time course of imaging. So then just by looking at this heat map, we saw that there are these hotspots which are shown here by red and dark red colors in the dividing cells. And we did not see similar hotspots in the non-dividing cells. So if you look at one example trace from each group, so here in purple was a cell that divided during our imaging time course, and we see this spike in the optical redox ratio, whereas we don't see similar spike in the non-dividing cell. So we did that for both HeLa and MCF10A cells, and we found similar hotspots for both of these cell lines. So to quantify that, to quantify the difference between the dividing and non-dividing cells, we normalized the redox ratio for each cell with its minimum, and then we calculated the maximum change. So here are the results. So we found that, in fact, the dividing cells had a higher change in optical redox ratio compared to the non-dividing cells, and this was significant. And this trend was similar for both the cell lines. So then our next question was, where does this peak in optical redox ratio occur, whether it's related to a certain cell cycle phase or whether it's just random? So to do that, we took advantage of the fact that the cells actually change its morphology, especially when the cell is in G2 phase and it's going into M phase. It rounds up, it becomes like a ball, and also it's at a different focus. And then right before cytokinesis, it becomes this H-shaped cell. And then, of course, we know the cells have divided because we can see two daughter cells. So now we can associate the redox ratio with the different cell cycle phases looking at the morphology. So when we aligned it with the morphology, very interestingly, we see that this increase always corresponds to the M phase. So whenever the cells round out for M phase, there is this increase in redox. And then as the cell goes through mitosis and divides, and then during early G1 phase, the redox ratio decreases. So then, of course, we wanted to make sure that this is occurring due to change in metabolism. We performed fluorescence lifetime imaging of dividing cells. So here is an example video where it's color coded with the lifetime. And this particular experiment, we followed the cells for 25 hours and then similar imaging parameters. So again, we can do single cell analysis and we can use the morphology to figure out the cell cycle phase. And in fact, we saw that the NADH lifetime actually decreases when the cell enters M phase here. So this means when the cell is in M phase, there is more free NADH in the cell. So then when we compared both optical redox ratio and NADH lifetime, we see that they beautifully align and they beautifully correlate with the M phase of the cell cycle. So far, all the experiments, we did it on unsynchronized cells. So we didn't add any other compound to synchronize the cell cycle phases of the different cell in our dish. But then we wanted to make sure that when we add different cell cycle arrest drugs, that we see similar results. So we used many different commonly used drugs here, but I will show results of a few of the important ones. So first, let's look at Taxol. So this is a chemotherapy agent, but it arrests the cells at M phase. So here we can see in our control group, most of the cells are in interphase, which can be seen because they're attached and spread out. And there were a few M phase cells. But then in the Taxol group, we found much more M phase cells, which can be identified by their circular morphology. So then if we quantify the results, we found that the optical redox ratio increases for the M phase cells and the lifetime decreases for the M phase cells. And this corresponds to our previous observation of the unsynchronized cells. So this is another compound, nocodazole, which would also arrest the cells in M phase. And we saw similar results in both NADH lifetime and optical redox ratio. And we also confirmed these results by doing biochemical assays with nocodazole arrested cells, and they match our optical redox ratio and lifetime. And then this is the third compound where we did double thymidine block, and this would actually arrest the cells at G1S phase. And as expected, we do not see a difference in both lifetime and redox ratio, and also in our biochemical assays between control and treated group. Lastly, we also looked at mitochondrial membrane potential during mitosis. So to do that, we use the dye TMRE, which is a commonly used mitochondrial membrane potential dye. And this time we looked at synchronized cells and we imaged for 4 hours. And these are the imaging parameters. So basically what we found is here in the red graph is the TMRE intensity. And very interestingly, we also see an increase in the TMRE intensity in the dividing cell. Now, again, we can match that with the phase of the dividing cell. For that, here I have calculated a morphological parameter called eccentricity. So eccentricity will basically tell us how circular a shape is. So basically, as the cell becomes more circular, the eccentricity value will decrease. So that's why I have plotted inverse eccentricity. So basically, the blue graph tells us when the cell enters M phase because it becomes more circular. And then to our surprise, we see that the TMRE signal actually increases or rises before the the cell would enter M phase. This means the cells are become more hyperpolarized before entering M phase. And then we wanted to compute the lag time between the two graphs. For that, we did cross correlation. And we see that more or less around the TMRE signal will start increasing 15 minutes before the cell enters M phase. So from this observation, we can conclude that the cells actually first become, the mitochondria of the cells become hyperpolarized first, and then that will drive the change in metabolism that we see in terms of increase in redox ratio and decrease in NADH lifetime. Okay, so to conclude the first part of my talk, in this work, we observed optical metabolic changes along the cell cycle in unsynchronized cells. And then the high temporal resolution of lifetime imaging allowed us to quantify the metabolic changes at M phase at single cell level. We found that the cell becomes more reduced when it enters mitosis. And this change in redox is driven by the mitochondrial membrane potential. Lastly, we also looked at other cell cycle phases, especially the S phase, but for that we had to use some cell cycle markers. So here, for example, we used Fuji, which is shown here in red, and then NADH in green. So we are still working on that, and for the interest of time, I don't want to go into details of that, but we do see changes in metabolism in other cell cycle phases too. All right, and then I will quickly talk about our work in the embryonic cell division. So for this, we looked at C. elegans, C. elegans embryo, mainly because these embryos are transparent and they are small, and that's why they provide excellent samples for imaging-based studies. And we just look at the first, second, and third divisions. And then for preparation, we could image them live. So we extracted them from the worm, and then we imaged them on an agarose pad through the cover slip. These are the parameters here. My video is not playing. I will try to play the video. There we go. And then we made sure the laser powers were less than 4 milliwatts. And then we performed lifetime estimation for single cells. So this is a representative lifetime image of a cell of an embryo going from single cell stage to 4 cell stage. And when we quantified the result, we actually found this decreasing trend in NADH lifetime from 1 to 4 cell stage. However, this trend became more significant when we normalized each embryo to itself. So this tells us that the embryo's metabolism might vary between embryos, and normalizing to individual embryos is a better representation of embryogenesis. We also quantified single cells, and we can look at the traces of NADH lifetime. We found differences between the first P0, which is the first cell, and then compared to the 4 cell stages. And then if we look at individual differences, there were a significant difference between AB and P1, which is basically the 2 cells in the 2 cell stage. And this difference was maintained at all the time points for all the embryos that we imaged. Next, we wanted to look at what happens during cell division. So here we are looking at the division of AB. So basically, we compared the differences in NADH lifetime between AB and AB div. So AB div is right before it divides. So we can identify that by this elongated shape. And we saw that, in fact, AB div had a lower NADH lifetime compared to AB. Similarly, we looked at the third cell division, where we compare P1 and P1 div. And again, we saw a decrease in NADPH lifetime between these 2 cells. And this is very interesting because this decrease in NADH lifetime actually matches our somatic cell divisions, where we saw a decrease in NADPH lifetime during mitosis. And then we also correlated the differences between the AB div and P1 div to quantify whether this difference is at embryo level. So this correlation plot is comparing the differences in lifetime between AB and AB div and P1 div. we found that they actually linearly correlate with a p-value of 0.02 and a correlation of 0.65. So this suggests that there is moderate metabolic correlation between the two division events at embryo level. Okay, to conclude the second part of my talk, here we perform label-free metabolic imaging of live C. elegans embryo during early embryonic cell division. We found that metabolic fluctuations within the same occurs within the same embryo at different phases. And then we found that there is a decrease in NADH lifetime at cell division, which was similar to our somatic cell division studies.